This module is on membrane structure. We're going to focus on a few experiments that led us to the understanding that the typical membrane of cells is a fluid mosaic. Well, first of all, the basic structure of membranes is that of a phospholipid bilayer, incorporating a number of other lipid molecules as well. The characteristics of membrane lipids is that they are amphipathic. That's the basis for a stable phospholipid bilayer. Amphipathic just means these are molecules that have a hydrophilic region at one end and a hydrophobic region. In the case of phosphatidylcholine and phosphatidylserine, which are two common phospholipids, you can see the hydrophobic end is, of course, the fatty acid tail. And the hydrophilic region can vary, but typically contains charged groups like nitrogens. Here you see nitrogens or even phosphates. Phosphatidylserine, phosphatidylcholine actually have both positively charged nitrogens, amino groups, as well as phosphates. We tend to think of cholesterol as being a health hazard, right? It deposits in coronary arteries and causes myocardial infarction, which means a restriction of blood flow, which leads to heart attacks. But cholesterol is, of course, important for a lot of cellular events, including the synthesis of steroid hormones, but it's also an important part of cellular membranes. And here's a glycolipid. In this case, it's a galactocerebroside, so it's an important brain cell lipid, but all cells have glycolipids, lipids with a sugar attached rather than a phosphate group. If you isolate pure phospholipids from cells or buy them off the shelf, you can mix them and you can cause them to form spontaneous phospholipid bilayer vesicles called liposomes. And here's a picture from the electron microscope of actual liposomes made in a clear liquid, and the cartoon next to them emphasizes the phospholipid bilayer structure of liposomes. These are interesting structures for many reasons. They are used as medication delivery vehicles, or at least the idea is that we'd like to use them that way. Create the right kind of liposome, put the right kind of medicine in to the liposome, inject it into the circulation, and the liposome, if it's made of phospholipids that are typical, say, of a target cell membrane, it is likely that the liposome's membrane, the phospholipid bilayer, will fuse with the cell membrane, thereby allowing the entry of its contents into the cell, substances which would otherwise not pass across a membrane. You may recall that when we discussed crystal membrane function in the mitochondrion, one of the functions that was investigated was which of the complexes that you could isolate from a crystal membrane actually function as a proton pump. You may recall that we isolated crystal membrane, broke it down into phospholipids, threw away the proteins, and then in a separate exercise isolated one or the other of the three major complexes that transfer electron. And we took each of those complexes in turn and mixed them with those phospholipids and created a phospholipid vesicle, but containing only one of the crystal membrane electron transport complexes, and used that structure to demonstrate that such complexes can pump protons. So we've seen liposomes before. You'll recall this from Bioscience 150, I hope. Membranes are phospholipid bilayers in which are embedded many proteins. We say a mosaic of proteins. And so here we have a classic cartoon of membranes. If you look closely, you can see that in an electron microscope picture of a membrane, we see what's called a trilamellar structure, which simply means three layers: a dark layer, a light layer, and another dark layer. Early attempts to interpret what that might mean led to various models for membranes until finally the fluid mosaic model was demonstrated by experiments I'm about to show you. We'll be spending much time talking about the plasma membrane in the next few modules, but all of the membranes in a cell, whether eukaryotic or prokaryotic, are phospholipid bilayers embedded with this mosaic of proteins. Of course, what makes one membrane different from another is which proteins are embedded, and to some extent which phospholipids are used in making the membrane. The flashing blue box surrounds a region of proteins in a membrane, and you can ask yourself, what properties does that part of each of these two proteins have to have in order to be stably incorporated into a phospholipid bilayer membrane? So think about that. Let's take a look at some different cellular membranes and see how much protein, how much lipid, and how much carbohydrate they contain. Well, here's a plasma membrane. It'll differ from one kind of cell to another, but generally about 50% of the membrane is protein. Lipid, in the form of phospholipids, ranges again from low 40s to about 50%, and a small portion is carbohydrate. It's actually sugar, oligosaccharides, glycoside-linked short sugar chains. Let's take a look at the mitochondrial membrane. There are no sugars, for one thing. You will find that a characteristic of the plasma membrane or cell membrane of cells is that they are sugar-coated. No other membranes in the cell, with the exception of the endomembrane system membranes, has sugars. And endomembrane system membranes, this would be the RER, right, rough endoplasmic reticulum, Golgi vesicles, and so on, they can be sugar-coated on the inside, but that's because those membranes actually end up as plasma membrane when secretion vesicles exocytose and fuse their membranes with plasma membrane. So you will see that the other kinds of membranes I'm going to show you don't really have sugar. So the mitochondrial membrane has no carbohydrate. It's made up principally of protein and lipid. And look, 76% of this mitochondrial membrane is protein. It is rich in the proteins of the electron transport complexes. It concentrates those proteins there to enable efficient respiration. Only 24% of this membrane is lipid. The chloroplast also has an electron transport system that's part of the photosynthetic apparatus. It is on the thylakoid membrane, so the thylakoid membranes of chloroplasts might be expected to be similarly rich in proteins and poor in lipids, and indeed that is the case. Here's a membrane you may have heard of. This is myelin membrane. It is found in or around nerve cells. Look at this. It's only 18% protein and 79% lipid. Now, lipids, being unable to interact with water, are very good insulators, that is, electrical insulators, and you may recall that neurons, nerve cells, are in the business of conducting neural impulses, which are essentially electrical impulses. So many nerves are surrounded by lipid-rich myelin membrane, which insulates the nerves, especially the long process called the axon, against dissipation of the nerve impulse. Again, the nerve impulse being an electrical charge. Let's look at the structure and function now of a typical plasma membrane. We're really focusing on eukaryotic cells here. I told you cells are sugar-coated, and by that I mean there are covalently bound oligosaccharides shown in this illustration as blue, looks like hexagons, I guess, if you look very closely. And these are glycoside-linked sugars, and they are covalently attached to the proteins embedded in the membrane, proteins being shown in green. So we have the phospholipid bilayer, we have the proteins embedded in the membrane, and we have the sugars that are covalently attached on the external surface. It's not stated here, but this is, the sugars are on the outside of the cell. So we say the cell is sugar-coated. We refer to the sugar-coated cell surface as the glycocalyx. We have several kinds of structures that are sugar-coated or that have sugars attached. There are short oligosaccharide chains attached to proteins, and these are glycoproteins. And then we have structures called proteoglycans, which are still transmembrane proteins, that is, proteins that pass through the entire phospholipid bilayer, but have longer polysaccharide chains attached. And the fact that these are much longer polysaccharide chains, that is, glycoside-linked sugar chains, differentiates them from glycoproteins. Also shown but not labeled are phospholipids to which sugars have been attached, and these are glycolipids, and we saw an example before. It says on the bottom of a slide, different regions of the cell membrane can have different functions. I alluded to that earlier. And here's the picture that was supposed to show up first. This is a cell, any cell. Think of it as a blood cell. It has multiple functions based on the proteins that are embedded in the membrane. So some proteins are receptors for receiving chemical signal information. That's number one on this slide. Some of the proteins are required in the case of, say, an amoeboid cell in order to get movement and enlargement of a cell in order to change its shape. So, for example, a phagocyte will change its shape as it engulfs a bacterium in your bloodstream. And then there are, of course, all these proteins which are involved in transport, either import or export of molecules. Here, in this kind of cell, you might expect to find such proteins pretty well evenly diffused around the surface of the cell membrane. But now, as it says down below, different regions of cell membranes can actually have different functions. Well, we will take a look at that in a few moments. Let's talk about the fluid mosaic model. It used to be called the fluid mosaic theory of membrane structure. You can take purified membranes or even whole cells, freeze them in ice, go get a hammer and chisel, and tap at the frozen ice block containing the membranes, and what will happen is the membranes will fracture along the line of least resistance, the interaction of the hydrophobic fatty acid tails of the phospholipids. So you will split the phospholipid bilayer into two halves. And when you do that, there's a cartoon from another textbook, and then there's a cartoon that I drew for you. And the idea is that if you split the membrane in half, some of the proteins will travel or stick with one half of the phospholipid bilayer, some with the other. I've drawn it so that they're all on one side, which is typically what you see when you actually look in the electron microscope. And what you can see in both illustrations is that if there's a protein stuck to one side of the phospholipid bilayer, because it was frozen, nothing had a chance to shift, and you can actually see a groove or a hole even where that same protein was embedded in the other side of the phospholipid bilayer. This kind of electron microscopy tells us that there are indeed proteins embedded in the phospholipid bilayer, that they are not, for example, sitting on either side of the membrane that is interacting, say, just with the phosphate heads of the phospholipids, but they're actually embedded and they stick all the way through a membrane. So all the ones shown here, with one exception, called a peripheral membrane protein, are called transmembrane or integral membrane proteins because they span the entire phospholipid bilayer. The illustration shows the oligosaccharides, and so without my telling you, you should be able to identify what's outside and what's inside relative to this bit of plasma membrane. The glycoside-linked sugars in the glycopeptides, and in one case a glycolipid, must be on the outside of the cell. Membranes are asymmetric. They're asymmetric, for example, because the sugars are only on one side of the membrane. So let's take a look at how one demonstrates this membrane asymmetry, structural asymmetry. It is possible to stick ferritin, now that's an iron-binding protein. There are small amounts of iron in living systems, including ourselves, and we would not be wanting to have little iron filings floating around in our blood, so the iron ions are bound to a protein called ferritin for delivery to wherever they have to go. If you stick ferritin onto a lectin, that's a plant protein that binds sugars, there's a lectin called ConA, concanavalin A. We'll see it spelled out in just a moment. It is attached to, we attach it to iron-bound ferritin, and on the other side of the concanavalin A is a sugar recognition site. So what lectins like concanavalin A can do is recognize sugars and bind to them wherever they find them. So we take a structure like this, a tube full of this stuff, and we bathe cross-sections of cells to this ferritin-tagged concanavalin A, or ConA for short. Then we look at what we've got in the electron microscope, and here's a cross-section of a cell shown first in low power and then in a smaller inset in high power. The electrons that are beaming in an electron microscope cannot pass through iron, so we say that iron is electron dense. In an electron micrograph, bits of real iron are going to appear as little black dots, and the little black dots then are iron that's attached to ferritin, which is in turn attached to ConA, which, as you can imagine, is now attached to the glycocalyx, to the sugars on the cell surface of this cell. And if you look carefully, you can see that all of the black dots are on the outside of the cell, not on the inside, and the inset shows some of these black dots in much higher magnification. If you look closely, you can see the trilamellar appearance of membranes in the electron microscope. Remember, you don't actually see phospholipids with their fatty acid tails and their phosphate hydrophilic heads. You can't see that in an electron microscope. What you see is this trilamellar structure, this sandwich of a dark line, a light line, and a dark line. And you can actually see that pretty clearly on the left side of the inset. But again, the iron particles are bound only to the outside of the cell. So that means that sugars are only found on the outside or extracellular surface of plasma membranes. Here's another experiment that demonstrates membrane asymmetry. And to explain this, I have to go a little bit into electrophoresis, which we'll be talking about in several contexts. So let's have a quick general explanation of electrophoresis. The setup is shown here with a chamber above and a chamber below with a power supply. This is a direct current or DC power supply. And we have an anodal chamber, which is the positively charged chamber, and a cathodal chamber up top. The proteins have been dissolved in solutions such that their net charge in solution, their net overall charge, is going to be negative. So when we turn the power on, when we apply a charge across a slab of gel, it looks like clear jello, the proteins that are in this electrophoretic gel will move towards the anode because they tend to be negatively charged. We're going to separate these proteins based on size. So when we turn the juice on, there is movement towards the anodal buffer chamber. Now what you saw moving was a blue dye that we threw in to the protein solution so that we could follow the electrophoresis until the blue dye reached the bottom of the gel, and then we turn it off. The blue dye has been chosen so that it moves faster than any proteins that we might have in the solution. So by stopping the electrophoresis when the blue dye reaches the bottom of the gel, we can see all of the proteins that have now separated based on differences in size, and we haven't lost any by having them run off the gel, for example, into the anodal solution. So the wells originally contained a protein solution with this marker dye. We take the gel out of this apparatus. We stain the gel with a colorimetric dye, that is, a dye that recognizes proteins generally and turns them colored, in this case a kind of a light blue. And so what you now are looking at are bands of proteins. Each band represents one or more proteins that has migrated at a rate that is proportional in some way to the size, that is to say, the length of the polypeptide. Larger proteins move more slowly, so they'll be nearer the wells where they started. Smaller proteins move more rapidly. Now let's take a look at how this technique can be used to show membrane asymmetry. So here's a bit of phospholipid bilayer, and there are several proteins associated with this bit of membrane. On the outside, we see the little hairy structures on proteins 2 and 3, which are the oligosaccharides of the glycocalyx. But we have several different proteins. We have 3 transmembrane proteins. These are proteins 2, 3, and 4. And on the inside surface, the cytoplasmic side of the cell, there are 2 additional proteins, numbers 1 and 5, which are peripheral proteins because they bind to the membrane either by binding directly to the heads of some phospholipids, as happens with 5, or they interact with integral or membrane-spanning proteins. That would be protein number 1, a peripheral protein that recognizes and binds to some portion of protein 4. Now that's an asymmetric membrane, isn't it? It has different components facing inside and out. Let's take a look at how we can test that electrophoretically. We can treat the whole cell first with a proteolytic enzyme. This is an enzyme that will digest proteins and polypeptides down to their amino acids. Trypsin is an example of one of these, and it's one of the pancreatic digestive enzymes that does just that, digests the proteins in your diet. First thing we have to do is extract the proteins from untreated cells and look at them on electrophoretic gel, and here we see all our 5 proteins differ in size and therefore separate on electrophoresis, and so we have a cartoon of a gel with 5 different bands. The smallest protein is number 1 in terms of the picture, and so we would expect it to move fastest or towards the bottom of the gel ahead of all other proteins. It looks like protein 3 is the largest one, so it should be the slowest moving protein in the gel, and so that's what the cartoon says. So these are representative of the full size of the proteins you see here. Now let's treat this same cell with trypsin. So we'll add trypsin to intact cells, and then we'll extract the membrane protein. So the cartoon you are now looking at is cells that have been treated with trypsin, intact cells that have been treated with trypsin, and the trypsin has digested parts of proteins 2 and 3, because that's all that's accessible to the trypsin in the outside environment of these cells. If we now extract these proteins and run them on a gel, we will see a gel that looks like the one that just appeared a second ago. Protein 3, remember, it has a chunk of carbohydrates and proteins on the external surface. After trypsin digestion, that's been lost. After digestion, we would expect band 3 or protein 3 to be somewhat smaller and move faster, and indeed it does. And so you see protein 3 in the second gel has moved a little faster because it's lost some of itself. It's been partially digested. And likewise, protein 2 should also move faster in the gel, and indeed it does. So in the second gel, we no longer see the original protein 3 or the original protein 2, but we do see 2 smaller proteins that have been derived from the originals by digesting intact cells. Now, it's also possible to chemically poke holes in cell membranes and allow the trypsin to actually get inside the cell so that the trypsin can digest not only what's outside the cell but also what's inside the cell. So we've added trypsin to a permeabilized cell of this sort, and what you expect to happen is shown in the cartoon. You should digest all of the protein components that are either on the inside cytoplasmic surface or the outside aqueous environmental surface leaving behind only those portions of the protein that actually span the membrane. In this illustration, you would expect to be left with proteins 2, 3, and 4. Proteins 1 and 5 will be digested in their entirety. And what's left of proteins 2, 3, and 4 look, at least in the cartoon, like they should be about the same size. And indeed, if you ran a gel, once again, the peptides remaining in the membrane after permeabilizing the cells and digesting with trypsin are proteins 2, 3, and 4, all moving to the same position, that of the transmembrane portion of each protein in the cell membrane. This is an electrophoretic way of demonstrating that the membranes of cell membranes are asymmetric. Okay, so we know that membranes are a protein mosaic, that the protein mosaic is asymmetric, and we also know that it is a fluid mosaic, that is, the proteins are not fixed in place in the membrane, unable to move, but in fact they can move. And this is an experiment. 2 cells, a human and a mouse cell, can be fused, and because the mouse and the human cell have different proteins, it is possible through the techniques described here after the fusion to watch the proteins of the mouse cell move into the region of the human cell membrane and vice versa. The technique here is a fluorescence microscopy technique based on making antibodies to mouse cell surface proteins and human cell surface proteins, and then tagging each of them with a different fluorescent dye that will fluoresce a different color, represented here by red and green. And what you see in an actual experiment is you can see live cells fusing, showing, say, red fluorescence on one side and yellow fluorescence on the other, and then over time the red and yellow fluorescences, if you will, mix, because the membrane proteins that are bound to those antibodies are moving laterally in the membrane and eventually diffusing throughout the entire membrane of this fused pair of cells. Here's another experiment that was done to demonstrate fluidity. So I mentioned concanavalin A before. It's a lectin, plant lectin, a protein that can bind cell surface carbohydrates, and that capacity was used in this experiment. Here, the con A was tagged with a fluorescent marker so that you could visualize the con A in the fluorescence microscope. The cartoon shows glycocalyx oligosaccharide coated proteins in green. So the cell is this circle coated with sugars, and the con A is this reddish crescent shaped molecule. Each con A molecule can actually bind more than a few sugar residues. So lectins are fairly large proteins with a number of different binding sites for sugars. So we have fluorescent tagged con A, and what happens when we mix that with the cells? Watch what happens. First they bind, and then after the binding, because each con A can attract and bind additional sugars, some of the proteins in the membrane move and stick to the con A that bound initially, say, to only 1 or 2 of glycoprotein. The result in the fluorescence microscope is to create patches of fluorescence around these cells, and that's in fact called patching. Sometimes it's called capping. So the clear implication is that protein components of the membrane can move along the surface of the membrane, that they are fluid. All right. So the membrane is a fluid mosaic. Fluidity depends on a number of factors. Clearly, membranes are more fluid at higher temperatures simply because greater thermal motion means that the phospholipids can expand and contract and bump into each other more, so higher temperature leads to greater fluidity. The degree of fatty acid saturation in the phospholipids also affects fluidity, and in this picture of a phospholipid bilayer, you can see that the kinks introduced into the fatty acids by double bonds in unsaturated fatty acids increases the spacing between the phospholipid tails, and if you push apart the phospholipids, then they will tend to be more fluid. That is why more unsaturated fatty acids lead to greater fluidity. Cholesterol, on the other hand, which is a major component of membranes, actually stiffens the membranes, and it does so by filling in the gaps between phospholipids, between the unsaturated phospholipid fatty acid tails in particular, and that results in a greater rigidity or inflexibility of the membrane, that is to say, a less fluid membrane. Now this has interesting consequences. You know that cellular life exists all over this planet, from the tropics all the way to under the polar ice caps. And we're talking here not about humans that are capable of living in all these places, but of cold-blooded animals. animals that don't maintain very high body temperatures necessarily, much above ambient, much above the temperature of their surroundings. So we have tropical fish, for example, and we have fish that swim under the Arctic ice. Think about how the membranes of the cells of these fish might differ in order to guarantee that there will be the right amount of fluidity to allow proper membrane function. Okay, early on I said that there are many functions mediated by the various proteins embedded in cells, including recognizing molecules on the external environment. They can recognize other cells, and if they do that, they can know which cells to associate with to form tissues, and so on. This is two half cells and one whole cell of an epithelial layer of the sort that might line your gut, for example, or line your blood vessels. Such epithelial cells are actually divided into membrane domains so that certain proteins are found only in one region of the membrane and other proteins in other regions. The cartoon demonstrates this by talking about proteins that are on the apical side of the cells, the apical side of the epithelial layer. In a blood vessel, these cells would have proteins that interact with the circulating hormone and nutrients. These would be on the apical layer, so the proteins on the cell surface on that side would be designed to recognize hormones or recognize nutrients and know to bind them and take them up. So the membranes of adjacent cells in this layer would have different proteins that are there to enable the cells to communicate or to stick together, as in the example of this thing called a tight junction. We did look at cell junctions briefly in 150. Perhaps you've looked at them more closely in other courses. We'll talk about cell junctions and how cell proteins participate in actually holding cells together, and obviously that's a domain of membranes as well. And finally, on the bottom side, the cells of an epithelial layer are actually attached to something, right? They're attached to the walls of the blood vessel, generically called the basal lamina, and obviously different proteins are required to effect that attachment. So, different proteins in membranes can be confined to different regions, in part by how the cells interact with one another and create barriers to the movement of these proteins. So a tight junction, for example, is a barrier to the movement of the proteins here, in this case called proteins A and B. A is on the apical surface and can't get into the middle, the lateral plasma membrane as it's called here, and those proteins B, which are on the lateral plasma membrane surface, likewise can't find themselves up in the apical surface. So the right proteins are placed in the right location on membranes. So that brings us to the end of a module on membrane structure.